Okay, so today in the last class, I asked you to go and read about C3, C4 plants. Did you do that? And someone please keep talking because with my video not on,
Yes,
I just rely on your voice. Okay, so
it's
please
okay. Me and
discuss.
Mariam will talk.
Yes, so I need someone to equally save the day for us so that we can continue with the class. Okay. Okay, cool. Can you see the screen that I'm sharing?
Yes, sir.
Okay, very good. So I asked you to read about C3, C4 plants because we started with nitrogen cycle in the last class, right? There are still certain details about the nitrogen cycle. The overall of the nitrogen cycle I told you in the last class, but that detail we have to study it, and also the oxygen cycle. And we know that plants utilize oxygen in the process of photosynthesis. So we first will venture into photosynthesis and we'll understand all the chemistry and the biology behind photosynthesis, and then we'll complete both the cycles together. Okay. And in this chapter, we'll be utilizing C3 and C4 plants, which I asked you to read. So did you get time to read on that? Yes or no?
Sir, I got disconnected last time, so I don't know what homework you gave.
Oh, okay. Okay, so let's talk a fresh topic today, which is photosynthesis. And I expect all three of you to discuss with me because photosynthesis is very important topic, both from entrance and from board point of view. And it's also the topic from which lots and lots of logical questions can be made, not just factual, right? Like in the case of mineral nutrition, there was a lot of facts to remember, correct? Photosynthesis has a lot of concepts. And then we'll go on to study respiration in plants, which is more or less, it's the cellular respiration. So it's more or less same for all living systems. But photosynthesis is something very, I'll not say exclusive, but yes, plants are major photosynthesizers apart after photosynthetic bacteria and algae, of course. So photosynthesis in plants we'll talk about today. So what is photosynthesis? As the word suggests, photo and synthesis. What do you think it means? We have discussed quite some things about photosynthesis. In today's class, I'll go into details. So let's first cover up the surface quickly. Photosynthesis. Photo means?
Light.
Light. Perfect. And synthesis is?
Breakdown.
Breakdown
No, that's not. Synthesis is making up. Synthesize something. Breakdown is kinesis.
Is it making up of food?
What it is?
Hello, am I audible?
Yes, you're audible now.
Yeah, is it making up of foods and all?
It's making. Synthesis is making or synthesizing something. Kinesis is dividing, and then lysis is breaking down. Okay. Lysis, synthesis, and kinesis. Don't get confused. I think you should write it.
So
in
can
your
you repeat
terminal.
for kinesis? Can you repeat
Yes.
for
I'll just write it here. So see.
Okay.
Synthesis
Sir,
is one.
what about genesis?
Yes, right. Good. Synthesis and genesis are associated with creating. So synthesis is making up or to make something. Genesis is to create something. Okay. It can also be used in the context of making. You understand? And then you have kinesis, which means dividing into two. Basically, it means dividing into two or break. It's also used for breaking sometimes. And then there is lysis. Lysis means breaking up, like cytolysis. So you will remember these four terms: synthesis, genesis, kinesis, and lysis.
Yes, sir.
Okay. So photosynthesis, we have Abhinav also joined. Hello, Abhinav. Good evening.
Good evening, sir.
Yes. So we are discussing photosynthesis. And so synthesis here is like making something. So in the presence of light, what are you making here?
Food.
Glucose.
Yes, the food. for the plant, which happens to be glucose, which is stored in the form of starch in plants, correct? Okay, so in the process of photosynthesis, plants utilize the light energy to do some chemical processes to make their food, because food is a chemical entity, and that chemical entity is glucose molecules, because any living cell utilizes glucose in respiration. So respiration is the next step that we'll study in the next chapter. So before we utilize glucose, we have to make glucose. We means plants have to make glucose, and we have to get it from somewhere. Like we are heterotrophs, so we have to get it from other food sources. But glucose has to be in the system so that it can be now broken down in cellular respiration. Is it clear? And in plants, it's stored in the form of starch, and also another form of starch in plants is cellulose. Remember cellulose? Plant cell wall is made up of cellulose.
Yes.
Yes. As humans, like animals, like we, which are not ruminant animals, like, so you know what is a ruminant animal, anyone? Let's discuss certain basics first. Ruminant.
Digest cellulose.
Yes, so basically the word ruminant comes from, there's nothing like cellulose
Rumen.
hint here. Yes, so they have an additional stomach called rumen, right? This is an additional stomach chamber that they have. So if you will see cattle grazing, and if you, how many of you have seen cattle grazing? And have you observed them grazing?
I have seen them. So
You have seen,
sometimes
Asha?
even they won't eat something, yet they will do those stuff. Like goats and cows. Right?
So
They will be grazing.
so when they are grazing, you will see that they are continuously just eating, you know, they are not chewing there. They're just
Yes.
biting the grass and swallowing it, because
Yes.
whatever time they have got and whatever resources they have got, they need to utilize it to the best. So, and basically they can digest cellulose from plants, as Mariam very rightly pointed out. So what they do, they hurriedly keep cutting through their teeth and swallowing that grass or that leaf or plant, whatever they are feeding on. Okay, and after some time you will see that they, when they sit, when they are back from grazing, they sit and they start bringing back that food into their mouth and start chewing it, and they chew a lot. Have you observed this? Cattle, when they are free and they're sitting, they keep chewing.
Yes,
So
I'm
if
also saying
you,
the same thing.
yeah, so if you in front of your grandparents or parents, you know, keep like you are chewing any gum or something, they sometimes tell you that why are you grazing like a cattle all the time? Like,
Yeah.
गाय भैंसों की तरह पगुरी क्यों करते हो? Like what paguri word is that word, if you understand? Okay.
Yes, sir.
Which tells us that they first store in, they just hurriedly store, and then they take it out and start chewing. By that time, there are certain enzymatic activity that happens in rumen that helps these animals to digest cellulose as well. So the word ruminant comes from rumen, which is the additional stomach in these ruminant animals. Okay. Now we are not ruminant, so clearly we cannot digest cellulose, right? We can only digest starch. So your potato is rich in starch. Okay, but a leaf, like any leaf, has both starch as well as cellulose. So cellulose is very difficult to digest for humans, so we basically don't digest it. It comes into the body and goes as it is. Starch component of it is utilized by us, but basically both are carbohydrates, and to digest starch we have amylase, and to digest cellulose, what should animals have? Cellulase, right? But in this chapter, the story is still the formation of glucose. So before we reach to photosynthesis and glucose and this whole story, let us first talk about how did we figured out that there is something called photosynthesis, and how did we figured out scientifically that what components are used in photosynthesis. So there are certain very interesting very early experiments which were done by scientists. Okay, and those are all science. History of science is very, very interesting. So those who are interested in history of science should read, and many of these experiments are also I think mentioned in your NCRT. But the most important person in this whole story is Joseph Presley. Now Joseph Presley, why he is Presley, yes, important, is because he's the person who for the first time discovered oxygen. Like, as a component of our atmosphere there is oxygen, so he discovered oxygen. Okay, and that you should remember, Joseph Presley. I hope you all know the difference between an invention and a discovery. Oftentimes people
Yes,
use the wrong word. Yes, Asha, what is a discovery and what is an invention? Mariam, yes.
Discovery is when you find something. Invention is when you like actually make and invent.
Yes, so invention is like you use raw materials to make something which was not naturally present in the environment, and discovery is just discovering something which was already present in the environment, right? So all the natural things or the chemistry or the elements can be discovered. Can you invent an element?
no.
Yes, if it is an artificial element, there are non naturally occurring elements also. Man-made synthetic elements they are called. Have you not heard of
So,
synthetic man-made elements?
Yes, sir.
Yes, like one is graph. There are many elements such like that. Like graphene is one substance that is man-made. We do not find it on the element table anywhere. Basically, it's made up of like carbon, but the way it is structured is different. Yes, anyone was saying something?
so actually, I was going to say, but I didn't say because sometimes I'm really saying wrong
Your
answers.
your voice is breaking for me. Is it breaking for
Difference
others as well, or
Yes, sir.
it is breaking, right? Aisha, your voice is breaking. Hello. I think Aisha is also suffering with some bandwidth issue, like me. Is my voice also breaking?
Is it?
No, right?
No, sir.
Okay, good. Yes, Aisha.
Hello.
Yeah, you're back.
Is it clear, sir?
Yes. What were you saying?
Hello, sir. Is it clear?
No, I think
Saying that-
it's still
Hello?
breaking for me. It's still breaking for me, Aisha.
Shall I
Can you type? Humans have one this very peculiar behavior that when the network starts lagging, we tend to get loud. You know, we don't realize that if we are loud, the network is not going to help, right? We also do it over phone. Like I have done it multiple times, unconscious, like say, like without a conscious mind, we do it. Like someone says the network is not good, we're like, what? Hello? Right? That's human behavior. Aisha, I think you should either type it out or wait for your network to stabilize. Okay, we'll come back to you. Yes, so she did, I think. I'll say something wrong. See, that inhibition you have to get rid of, and especially in my classes where I urge you to give answers, and I tell you that the worst that you can do is you can give a wrong answer, which is completely fine in my class, or it should be in any class, because unless and until you give wrong answers, you will not know what the right answer is, right? So, and you have to give wrong answer somewhere. Every student who's learning has to give wrong answers. Now you decide whether you want to give all the wrong answers in a classroom or you want to save some for your examinations. So where would you like to spend all your wrong answers? In the classroom, I suppose, right? Yes. So
is my voice okay now?
yes, it's fine now. So you give
Actually,
all your wrong answers here.
yeah, if you don't mind it.
See, I will not mind if you are giving all the right answers in the exam and all the wrong answers in my class. That's fine, because you know that what is wrong and what I should not write in the exam. Okay.
Yeah.
Okay, cool.
So I was going to say that, shall I say?
Yes,
Because
yes, yes,
that
yes.
discovery is something like recognizing that already exists for the first time, like that nobody has found.
Yes, but exactly, like it
Yeah.
should be
And
naturally present
and
in the
invention is
environment.
something that creating totally new with their own ideas and development.
Yes, using raw materials from the nature. That's
Yeah.
invention. Okay.
Yes, sir.
Yes, like ideas most of the times are placed in the category of invention. So idea has to be invented because there was nothing like that before, and that is one thing where we are not using raw materials from nature when we are inventing ideas. So theories, like theoretical ideas in theoretical physics or theoretical biology, are also called uh, categorized as inventions and can be patented. Okay, do you understand that?
Yes, sir.
Yeah. Hello, am I audible?
Yes, sir.
Yes,
Yep.
sir.
Perfect. So let's move forward. So Joseph Presley, you have to remember him for the discovery of oxygen in the atmosphere, and he did it in the late 18th century, somewhere around 1770s, like 1774, if I'm not wrong. He discovered this, that oxygen is there. Now, how did he discover that there is something called oxygen? And uh, like he named it oxygen, but what it basically does. So he figured out that there is something common in the atmosphere that both a plant system and a living system is utilizing. Okay. Do you understand? So there's something which is required for, first of all, so you must be knowing about these experiments where a candle was, like there was a candle lighted and it was covered with a glass lid. So after some time, what will happen?
It will stop burning.
Yes, you will see that the candle is extinguished, right? And why is
Yes,
this
sir.
happening? Like now we know the
Because
answer. Yes, because
no oxygen.
then why it was burning in the beginning?
Because there was oxygen available at that time,
So
and
what
then
happened to the oxygen?
it was used in burning.
Yes, so Presley got to understand, because he was not knowing the chemical dynamics at that point of time. So he just concluded that there was something in the air. By covering it with a lid, a glass lid, all he did not allow was, like through a glass heat can come in or go out, through a glass light can come in or go out, but through a glass air cannot come in or go out, right? So he prevented additional air from coming in, and whatever air was present already had something which got used up during the process of combustion. Correct? Is it clear?
Yes, sir.
Yes, sir.
Now, so this was first thing. Second thing, he also saw that if you keep any living thing, like let's say a mouse, okay, if you keep a mouse in the same glass lid, after some time you will see that the mouse also dies, right?
Yes.
And why is that? Why does that happen? Because of the same reason. Oxygen got, like it got utilized, it finished. And who utilized the oxygen? The living system. Like the mouse utilized the oxygen, correct? Which means something is common which is getting utilized both in combustion and when a living system is breathing in air, correct? We now know, after all this time and all this research, we now know that what we do with oxygen in our body, when we inhale oxygen, where do we utilize it?
In the cells,
In which process?
respiration,
Perfect. To do what?
metabolic activities,
Break down glucose,
breakdown.
oxidize glucose, correct? And when oxygen is used in a combustion process, you will know from your class 10th chemistry that that oxygen is also used to break down things, correct? This wax or what, this source of fuel was broken down by oxidizing it, correct? And CO2 is
Yes, sir.
released. Similarly, in our respiration process, we break down glucose and CO2 is released. So on a chemical level, it's similar kind of a reaction happening, but we didn't know this then. He also figured out that instead of a animal, if you keep a plant, okay, so there is a plant and there is a burning candle, okay. Now what will happen? This candle will extinguish, right? But will there be any difference this time? What do you think will happen? Like, you think logically, if you had to do this experiment for the first time, suppose I tell you nothing about the results and I ask you to do all these experiments. So there are something called thought experiments, which Einstein used to do. He did all sorts of thought experiments for his theory of relativity, because the experimental thing was not possible. So let's do a thought experiment. Suppose I give you a glass lid and I ask you to keep a plant along with a burning candle inside it. So you already have done this first experiment where you noted down the time. Let's say after time t, the candle extinguishes. And here also you got to know that after time t, there's something, the red dies. Now, if you keep a plant with a candle or a plant with a mouse, what will happen? What do you expect to happen logically?
It will still burn.
It will keep on burning forever? Will it keep burning forever? Hariom?
Sir, not forever, but like longer than without the plant.
Exactly. So very good thought experiment, that this burning of the candle or the flame of the candle will prolong, right? So we got what we can conclude, if it is t plus x in this case, so this x, by this x factor, this burning got prolonged. So what do we conclude? Anyone? Quickly. You're
Plant
just
gives oxygen and utilizes the CO2 released by the candle.
so plant is producing what candle is utilizing, correct? We yet have not named it, let's suppose, but we know
Yes,
that
sir.
whatever that candle is using to burn, plant is producing it, correct? Right.
Yes.
Yes. And if there is animal also, that animal will also survive for longer, correct? So whatever that animal is taking up to survive, the plant is producing that. So these experiments were done by Presley, and he found that he hypothesized. So write down Presley's experiment. Let me just, because this sometimes is asked in boards. Write down, Presley hypothesized that plants restore to the air. Presley hypothesized that plants restore to the air what breathing animals and burning candles take from it. okay, which he later identified as oxygen, Is it clear?
Yes, sir.
So we got to know the very first kind of hints that plant gives out oxygen from Presley's experiment. So we know that the first character of our play here, which is oxygen, is in the picture. Okay. Now, what we still don't know is that what happens, like this oxygen, which is produced by the plant, why it is produced and in which process it is produced, and when it is produced, what's happening, right? Something must be happening because of which plant is producing oxygen. Now here comes the second character. So in photosynthesis there are three things very important. First is sunlight, okay, second is oxygen, and the third is, in the raw materials, the third is what? What is the third one?
Water.
Yes, water. And the final product is glucose, right? So in the around mid 19th century, it was not known till then. So it took almost like 60-70 years, somewhere around the 1850s. There was another person called Julius von Sack. Julius von Sack. So he basically figured out that glucose is being produced by a plant. Okay, so when plants grow, glucose is produced. Now, how many of you have in school, in your primary classes, have done this experiment where you do the iodine test on the leaf to figure out that whether there is starch or not?
Sir, I did.
You did, Mariam? So would you like to explain that experiment for me, please? What did you do in this experiment? What do you remember doing?
Sir, we will cover the half part of the leaf.
Yes, with what?
With something that will avoid the sunlight going.
Yes, right. So something which is opaque, which should not be transparent. Very good, beautiful. So you cover half. Let me keep also drawing this for you. So you are saying that you took a leaf, right? And you did it like after breaking the leaf, or when the leaf was in the plant only? So this is a leaf, and half part of it you are covering basically with something that does not allow sunlight to go through it, right? Yes, Mariam.
Yes,
please.
sir.
Yes. Then?
And then the next day or something, we will put the starch on the leaf. And then something, we could see that photosynthesis was not done on the part where the paper was.
You did not put starch on the leaf. The test is for starch. What did you put?
Not starch, iodine.
Yes, the tincture of iodine you put.
Solution.
So what happens in the presence of starch? If there is starch, the tincture of iodine changes color, right?
Blue color?
Yes, something purplish, bluish purplish, right?
I don't remember, sir.
You don't, but you remember that there was a color change.
Yes, sir.
Okay, yeah, okay, that will suffice. So basically you covered the leaf with a black paper and expose the leaf to light, or you can do this experiment while the leaf is still attached to the plant. So you choose a leaf in the plant, you cover half of it with paper and leave it for a day. So the other half, which is exposed to sunlight, will keep doing photosynthesis, right? This is basically what is happening. And the part which is covered will not be able to do photosynthesis because light energy is not being captured in this area. Am I correct?
Yes, sir.
Yes. After that, what you will do? You will put tincture of iodine on the leaf, and this part of the leaf where photosynthesis was positive will show a change in color, right? Because iodine changes color when it reacts with starch. In this case, when there is photosynthesis negative, the color will not change, correct? This is what you
Yes,
recall,
sir.
Mariam? Yes. And this tells us that glucose or starch is being formed in the leaves, okay, through photosynthesis. Do you understand?
Yes, sir.
Yes. And it was also found that the later studies by Julius von Sachs also found that there is a factor called chlorophyll, which is present in all the green parts of the plant that allow this formation of glucose to happen. Is it clear? So he told chlorophyll, and these chlorophyll is present in the organelles which are called chloroplasts, correct?
Yes, sir.
Plastids, you all must have studied plastids, right?
Yes, sir.
Yes. So chloroplasts are within the plant cell. Now he found that the green part is where the glucose is made and that the glucose is stored as starch. So we got to know about glucose also. So there are two things now in picture: oxygen, glucose. We have yet not joined all the dots, but we know that glucose is released by the plant. Sorry, oxygen is released by the plant, glucose is synthesized by the plant under something, and we have chlorophyll, which are present in all the green parts, which allow this thing to happen in the presence of light. Now, what is there in sunlight? Let's come to the third thing. Sorry, just 1 second. Yes. Now, what is there in the light? Now, for this, there is another person you must be knowing about, who's called Engelmann. It's called T. Engelmann, or just Engelmann, you can write, because I remember just Engelmann. So this person here had a brilliant idea in his mind. So what he did a very interesting experiment where he used prism, because prism, by that, like by who first showed that light can be split, sunlight can be split into 7 different colors of Vibgyor? Our beloved Newton, the physicist. You know about Newton's splitting of light?
Yes, sir.
Yeah. So through a prism, what is prism? Everyone knows what is prism, or should I tell you? A prism is like a pyramid shaped special glass or lens through which, when light enters, so let's make a 2D of it. It's a 3D basically. So white light enters from one side and gets split into all its components when it comes out from the other face, correct? So prism basically splits all the wavelengths which are present in the white light. Now white light is a mixture of all the lights, that's why it's white. If something is white, it has all the wavelengths present. If something is black, there is nothing present. Is it clear?
Yes, sir.
Yes. So he used a prism to scatter the white light and detect and used a green algae, which is Cladophora. So the experiment was something like this. Let me draw it for you. So there will be a prism, okay, through which white light will get split, okay, into different components. Now there'll be a tube, a test tube, which will be containing Cladophora bacteria suspension. And in this experiment, what he will do is that bacteria accumulated. So these bacteria can do, Cladophora, which is a green algae, was placed in a suspension of aerobic bacteria. Now, what is an aerobic bacteria? First, let me make this thing clear. Aerobic and anaerobic bacteria. These are basics.
Bacteria.
Which bacteria?
True bacteria.
No, that is eubacteria, and eubacteria is true bacteria. Archaebacteria is not true
Yes,
bacteria.
sir.
Aerobic and anaerobic bacteria. Aerobic means which utilizes oxygen to produce energy. Okay, which means which can basically do the cellular respiration the way we do cellular respiration, which is oxygen dependent. Okay, that is aerobic. Anaerobic is which do not require oxygen. Oxygen is not required for their respiration. They use something else like sulfur or something. Is it clear?
Yes, sir.
So if aerobic bacteria utilize, so if a bacteria is aerobic, it can only survive in the presence of oxygen. Now in this test tube, everywhere cladophora is there. It's a suspension of cladophora. Cladophora is a green algae. Now by saying it green, I am just specifying that it has chlorophyll, right? Which means it can photosynthesize, correct? So cladophora will do photosynthesis. And since different tubes or different parts of the same tube, like you can do the experiment both the ways, but let's say different light wavelength, different colored light is falling on different tubes, let's say, or different part of the tube. So the cladophora which is present here, 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 and at all different places will do photosynthesis or not. So in the presence of this white light, plants are doing photosynthesis. Now Engelmann very ingeniously did split that white light into all components. Now under each component, he checked if photosynthesis is happening or not. And in the same suspension, along with cladophora, what is suspended is your aerobic bacteria. So how can you read out? Do you understand the experiment now? So in whichever part photosynthesis is maximum, oxygen will be produced the maximum, correct?
Yes, sir.
And since aerobic bacteria need oxygen to survive, they will accumulate in that part the maximum. Is it clear or not?
Yes, sir.
So if, let's say, starting from Vibgyor going towards violet, indigo, blue, green, yellow, orange, and red, there is a Vibgyor. And if we see that, for example, aerobic bacteria mainly accumulates in the region of blue and red. What does this mean? They are not accumulating in this region and they are not accumulating in this region. Most of aerobic bacteria are accumulating in the region where the cladophora or the green algae suspension is exposed to blue spectrum of the sunlight and the red spectrum of the sunlight. What does this mean? I have explained you the experiment. Now you have to just tell me the conclusion. What will you conclude? If you were Engelmann, what would you have concluded?
More photosynthesis will take place there.
Where?
At the red and blue.
Yes, which also means, remember I told you that chlorophyll is a substance that converts light energy into chemical energy, which means,
Yes, sir.
and I also told you when we were studying hydroponics, I told you that you can actually grow plants in the house because sunlight is also something which can be substituted for. And while telling you that, I told you that we just have to know that which particular wavelength of light excites the chlorophyll the most or activates the chlorophyll, correct? So we
Yes.
can just use that wavelength and make chlorophyll do what it does. We do not need the whole spectrum or the complete sunlight for that. And Engelmann found that the blue region and the red region of the spectrum are where photosynthesis happens the maximum. Is it clear? So you can just write it down. Photosynthesis is maximum, which means chlorophyll gets activated The chlorophyll which is present in green algae or in plants gets activated under which spectrum of the sunlight? Blue and red. Do you understand this?
Yes, sir.
Everyone understands this or not? The experiments, because based on these experiments, there will be questions. So those questions will be logical. So if anyone has any problem understanding, please tell me. Everyone understands that? This? Can you see the screen that I'm sharing? Has the screen changed for you or not?
No, sir.
No, okay, just wait 1 second. Let me bring it to the main screen. Okay. So, perfect. Okay, can you see now what I have pasted here?
Yes, sir.
Now, this is a figure from your NCERT. This figure is given in boards also and in entrances also, and questions are asked. Now, let's try to understand. Okay. Aisha, are you there?
Yes, sir. So
Okay.
actually in my home it was coming really bad, and from 10 minutes it was reconnecting. So I came to my Wi-Fi. Anyway,
Okay.
Is it
Okay, fine. Yes, it's
now?
fine. It's better now.
Okay.
So I think you missed
sir.
something. So just pay attention, okay?
Okay.
So I just told that Engelmann did an experiment where he did split white light. So he split white sunlight. We call it white because it's mixture of all the lights. So white sunlight into a spectrum, Vibgyor, violet, indigo, blue, green, yellow, orange, red. And he saw that photosynthesis was maximum in the blue and the red spectrum. And what was the readout? He used cladophora, which is a green algae, which contains chlorophyll and which will get excited by one or the other spectrum and do photosynthesis. And as a readout, there were aerobic bacteria accumulation. So aerobic bacteria are those bacteria which only survive where oxygen is present. Okay, so they respire aerobically. Now, he figured out that aerobic bacteria's accumulation was most in the blue and the red region of the light, which tells that photosynthesis was maximum there. That's why they are accumulating there, because oxygen will be released there. Do you understand?
Yes, sir.
Now, this is a figure from your NCERT. Just look at the figure A. This is what we have accumulated after all this study. So there are three major pigments, two chlorophyll A and chlorophyll B. Chlorophyll A is the major green pigment in plants. And then there is carotenoids. Carotenoids are the brownish reddish pigments. Carotenoids are present in carrots. And how do carrots look?
Orange.
Yes. What is we discussed one day, what is the actual color of carrot, the wild carrot?
Blue, black.
Bluish
Black.
black. Yes.
Yeah,
The actual wild carrot is bluish black, from which we humans have artificially selected and produce lots of varieties of it, because there are orange carrots, there are red carrots, there are yellow carrots, all sorts of carrots. It's the same like capsicum. You will see all different kinds of capsicum, correct? Orange, red, green, and many different kinds of cauliflowers varieties also. Broccoli, cabbage, cauliflower, all sorts. So carotenoids is non-green pigments in plants, which also helps in photosynthesis to some extent, but not very greatly. So let me teach you how to read the graphs. So on the y-axis here, it says that absorbance of light by chloroplast pigments. So the higher you go in y-axis, the more absorbance you are doing, right? The more light absorption is happening, correct? This is what it means. And on the x-axis, so this is x-axis and this is y-axis. If you go farther in the x-axis, it is the wavelength of light, starting from 400 to 700 nanometers. Can you see the here? It's common for all the three graphs. So let us just write 400 to 700. Okay, is it clear, everyone? Nanometers. And this is our visible spectrum as well. Do you appreciate this, everyone?
Yes,
Yes, sir.
Our visible spectrum is from 400 to 700. And somewhere around 400, between 400 to 500 will be your violet, indigo, blue, right? And green, yellow, orange, and red, correct? Vibgyor will
Yes,
go in
sir.
this series. So you see chlorophyll A shows the maximum peak somewhere around 460, right? 460, 470, which comes under blue, correct? This is also what Engelmann also saw in his experiments, that aerobic bacteria were more accumulated in the blue region. Is it clear, everyone?
Yes.
Yes. And again, the chlorophyll A. So this is the line. Let me use maybe red for chlorophyll. No, green. Green will be a good color for chlorophyll A. Let's use green for chlorophyll A. So this is chlorophyll A. Can you see this, what I'm drawing? Is it visible, the green one? Is this visible on the screen or not? People say yes or no. I'm drawing. Is it visible to you?
Yes, sir.
Yes. So you see this green for chlorophyll A. It shows a peak near blue at blue,
Yes,
and then there is a dip,
sir.
like, which means absorbance is not there. It was not there, or it was very low in the violet or indigo region, like this region. Then again, it's very low here, which is your green, yellow, orange region. And when it comes around red again, it again shows a spike, but this time this peak is smaller than what it was in blue, correct? So chlorophyll A shows two peaks, one in the blue region and one in the red region, which shows that chlorophyll A can absorb energy from two spectrums, blue spectrum and the red spectrum, around 460 nanometers wavelength and around 700 or 650 nanometers of wavelength. Is it clear, everyone? Is it clear to you? Say yes or no. Am I audible?
Yes, sir.
Yes. So is it clear to you?
Yes, sir.
But the amount of absorbance of light energy is maximum for the blue, and it's not that much for the red, like it's less for the red. So in which light photosynthesis will be maximum? In which spectrum the photosynthesis will be maximum? You tell me. The blue spectrum, correct? So this is a
Yes, sir.
question, it's a direct question. Chlorophyll A is most active or absorbs the light energy maximal in which spectrum, or which wavelength excites chlorophyll A the most? So what will you say?
The wavelength.
Yes, the wavelength for the blue spectrum of the visible light, which is around, write down, around 440 to 470 nanometers. 430 also, 430 to 470 is the range. This is where photosynthesis happens the maximal. Then if you provide red light spectrum, which is around more than 640 nanometers to 700, again a range, plants will also show photosynthesis in that spectrum, but less than what they will show in blue. Okay. Now let me ask you one question. Okay, so the first graph is clear to all of you?
Yes, sir.
Okay, let's go to the second one. Now this shows rate of photosynthesis. And how can you measure the rate of photosynthesis? So if you go again in the x-axis, it is telling you more photosynthesis. Sorry, y-axis, it will telling you more photosynthesis happening. And we measure that by amount of oxygen release. And in the x-axis, we see again the same, the wavelength of light. Now we know that absorption happens maximal for chlorophyll A in these two bands. So the photosynthesis will also be maximal in, so there is two peaks. One is this in the blue region and the other is this in the red region. Now the question is, here for chlorophyll A, there is a very stark difference in the peaks, but for the rate of photosynthesis, it is almost the same, correct? Why? Can anyone answer me? The answer lies in the above graph. Did you understand the question? Abhinav,
Sir, could you repeat?
the question is, for chlorophyll A, which I told you is the major pigment for photosynthesis, there are two peaks of maximum absorbance. One is in the blue region, one is in the red region, but there is a difference in the peak, correct? Can you see my cursor?
Yes, sir.
But when it comes to rate of photosynthesis, we see that there is no difference. Almost similar peaks are there. So the rate of photosynthesis, whatever it is in the blue region, it seems to be almost the same in the red region as well. So plant seems to do equally well in blue or red light, whereas chlorophyll A is not excited equally in blue and red light. Why? Why then photosynthesis rate is almost equal?
Because of other pigments helping.
Yes, right. Because of, use for chlorophyll B, not a blue. Which color should I use? Something different from blue. Let me use this one. Okay. Because there is another pigment called chlorophyll B here. Look at this one, which I'm drawing. This is chlorophyll B, the purple one, which I'm using. You see that B also shows a peak just before the blue, probably in the indigo region. And then it goes down. There is no absorbance. And then it again crops up in the red region. Correct? And in plants,
Yes,
all these pigments are present. It's not like there is only chlorophyll A or chlorophyll B. Chlorophyll B and chlorophyll A are present together along with carotenoids. Now for carotenoids, let me use something brownish or orangish. So this is your carotenoid. See, carotenoid shows two peaks, but very closely in the blue and the green region, right? These are the two peaks, and then there is no peak. Is it clear? So that's why you see that the maximum photosynthesis happens in this region, what I am covering here, the area under the graph. Can you see this? And then it happens in this region, right? It does not happen in this or beyond that, correct?
yes, sir.
And this is because of the pigments in the plant, and they together contribute to photosynthesis. So this third graph here is a summary.
Sir?
Where, yes, Aisha.
I'm really sorry. It's getting disconnecting again and again due to bad weather.
Okay, so, so you want me to repeat this last bit?
Please, yes, sir.
Yes, okay. So you see here, there are three colors that I have used because there are three major photosynthetic pigments in plants. The green one is chlorophyll A, the purple one is chlorophyll B, and this orange one is carotenoids. Is it clear?
Yes, sir.
Now you see that both or all three of this pigment has one peak which is near blue. You see there are peaks in blue region for all three.
Yes, sir.
For carotenoids, there is a peak in the green region as well. But after that, in the yellow or orange or like before red, there is no peak. And then in the red region, there is peak for chlorophyll A and chlorophyll B both. That's why when we see the rate of photosynthesis in plants, we see that the rate of photosynthesis increases in the blue region, right? This is the area under the curve, this one, and also in the red region. and the difference is not much because there are two pigments at work here. So these two pigments, the chlorophyll A and B in the red region, also makes plants able to photosynthesize in the red light almost equally well as they can photosynthesize in the blue light. Is it clear, Aisha? The question
Yes,
which was
sir.
asked in boards was that there is a great difference in the absorbance of chlorophyll A in the blue and the red light, red spectrum. See, there is a lot of difference, right? This peak is higher and this peak
Sir.
is very low. Yes. Anyone is asking anything?
No.
But even after this difference, like instead of this, even in spite of this difference, the rate of photosynthesis measured is same, almost similar in blue light as well as in the red light. Is it clear, everyone?
Yes.
Which is summarized here. Now you see this black line is for the rate of photosynthesis, and this is this gray line visible to you, everyone?
Yes, sir.
Yes. So this gray line is for combined absorption by all the pigments. So there are two peaks, distinct peaks in the red region and the blue region, and this peak is merging with the peak of photosynthesis. Clear?
Yes, sir.
So completely understood this graph. Is there any doubt? Please ask me, because this graph can be altered, so you have to remember this graph. Like if I make any alteration in this graph and ask you why this graph is wrong, or ask you question based on this graph, will you be able to answer, everyone?
Sir, could you summarize the third graph once again?
Yes, sure. So the first graph tells you about the absorption, like the absorbance by these three pigments in the so y-axis shows the absorbance and the x-axis shows the wavelength of lights in from 400 to 700, which is the visible spectrum, Vibgyor. Okay, and we see that for all the three photosynthetic pigments, which is chlorophyll, carotenoids, and chlorophyll B, there is a peak in the blue region, right? And then for chlorophyll A and chlorophyll B, there is a peak in the red region. In between, there is no peak. Is it clear?
Yes, sir.
The second graph just correlates, tries to explain the first graph by the rate of photosynthesis. Now on the y-axis, as you go higher, the photosynthetic rate is higher, and on the x-axis is same, the wavelength. And we see that the photosynthesis is also higher in the blue region and the red region, right? And then the third graph is the summary, showing like the peaks of absorption and the peaks of rate of photosynthesis both actually merge at the same point. Is it clear?
Yes,
in the
sir.
blue region and the red region. So will you be able to explain this result?
Yes, sir.
Yes, sir.
And this comes now, like after lots of decades of Engelmann's experiment, where he also figured out similar kind of thing, that blue and the red spectra of the light, of the sunlight, causes the maximum photosynthesis. And his readout was accumulation of aerobic bacteria in a suspension of cladophora. Is it clear?
Yes, sir.
Any doubt in any of the experiments right from the beginning, from Presley's, then to Julius von Sack, and then T. Engelmann? So these three names you have to remember. Joseph Presley first figured out oxygen, and also figured out that whatever a burning candle and a rat is taking away from air, plants are giving that to the air, right? Because of which, if you keep a burning candle in the presence of a plant covered by a glass lid, the burning time will increase by a factor of X, whatever it is. Is it clear, everyone? So oxygen
Yes,
came into picture. Then we moved to Julius von Sack, and Julius von Sack said that as plants grow, they also produce glucose. And that is happening because of the green parts of the plants which contains chlorophyll in chloroplasts. Yes, Aisha is asking, can you explain the second experiment? Is it this one, Aisha? What I'm talking about right now? Julius von Sack? Say yes or no. Yes, okay. So in the second experiment, the second character of this whole play, oxygen was figured out by Presley. Now we figured out that glucose is there. Now Julius von Sack figured out that glucose is produced by plants when they grow, and it's produced by the part of the plant which is green, which contains chlorophyll, which is present in chloroplasts organelles. Okay. Now you can also, how did he do that? The experiment is like sorry, if you take a leaf and you cover half part of that leaf with a black paper to not allow sunlight to fall on it, so basically you are not allowing photosynthesis to happen, right? And in the region where sunlight can still fall, photosynthesis is positive, it's happening. Now in this part, if you put iodine solution changes color in the presence of starch. Okay. Now in this part, the color change will happen. Whereas in this part where it's covered, the color change will not happen because chlorophyll are not allowed or they are not exposed to sunlight. Is it clear? But this tells us that glucose is produced because glucose is stored in plants in the form of starch, which can be tested here with this iodine solution change experiment. Is it clear, Aisha, now the second experiment? I think you lost connection. Yes. Okay. And then the third one is where the where E. Engelman actually took the third character of the play, which is sunlight. Okay, so we need oxygen, glucose, and sunlight. So what about sunlight? What is there in sunlight which makes sense? So he figured out that what he can do is he can split the sunlight into all its 7 components, the VIBGYOR, the visible spectrum, and then can see which spectra causes the highest or the maximum photosynthesis to happen. And we know that the readout of photosynthesis is oxygen from the very first experiment by Presley. And to see where is more oxygen, you can use aerobic bacteria where they will accumulate the most, oxygen will be the most there. Okay. And he also figured out that blue and red are the regions where most aerobic bacteria were accumulated, and hence it is the most photosynthetically active spectra. Now we know through research that it's true because chlorophyll A, carotenoids, and chlorophyll B, all the 3 pigments which causes photosynthesis, absorbs the most amount of light energy in the blue and the red spectra, where the photosynthesis is also the highest. Is it clear, everyone? So that is the summary. Completely clear? Or any doubt is there?
No, sir.
No doubt? Okay. Now take 2 minutes to summarize everything, then I'll move forward to the chemistry of it. Okay. Give me just 2 minutes. I'll take a quick water break. Okay. Okay. Do you allow me a water break or no?
Yes, sir.
Okay, cool. Thank you. Take 2 minutes and revise. I'll come back and start with the chemistry of it. Okay, hello. Hello, am I audible, everyone?
Yes,
Yes,
sir.
sir.
Yes, very good. So all is clear? Shall I move forward?
Yes, sir.
Okay, very good. So by the time the middle 19th century came, which mean around like 18, where till the time when Engelmann did his experiments. Now Engelmann, you should also write when he did around some 1860s, 1870s probably. Okay, and Julius von Zech was in 1850s. Yes. So by the middle or late middle 19th century, we got to know one thing. We got to know that plants can use light energy to make carbohydrates from CO2 and water. Is it clear? Do you understand
Yes,
the
sir.
sentence? That we know that plants are using light energy. Yes, plants are using light energy. They are using that light energy to produce glucose, which is a carbohydrate. And what raw materials they are using? CO2 from the atmosphere and water from the soil. Now, how to write this thing chemically? So write down write down this one line. The key features of plant photosynthesis, the key features of plant photosynthesis were known by the middle of 19th century. By the middle of 19th century. And just quick thing, like, you know, whenever the century is named, so whatever is the name of the century, the year has to be one before it. Like which century we are currently living in? We are living in which century, people?
21st.
Yes. And what is the year that we write? 20, right? Whatever is the year. So 2022 is going on, which is 21st century. If I say that something is 18th century, then the year will be 17 something. Okay, this is 18th century, because the first century, when it all started, the year was 0001. This is where the first century started, right? So first was 00. That's why everything is. Do you understand? Sometimes this century based questions, in when it has to be converted in years, students get confused. So I hope you don't get confused in this. Now write down. It was known that plants can utilize light energy to make or to synthesize carbohydrates from CO2 and water as raw materials. From CO2 and water as raw materials. And oxygen was evolved in the process, right? Plant was giving oxygen to the environment, which the burning candle was utilizing, correct? So all these things were clear by the middle of the 19th century. Now let's try to put it in equations. Now first thing, we know that the first raw material is CO2, and we know that there is water from the ground, and in the presence of light, because light itself is not doing anything. So it's not a reactant, it's a condition. So condition is always written either above or below the arrow. Okay. What is forming? A carbohydrate. Now, what is the formula for the basic carbohydrate? Chemistry people, 10th class chemistry, quick.
CH2.
What?
CH2.
CH2. CH2O. Right? This is the formula. So CH2O plus oxygen was evolved. Okay. Right. Now this CH2O, if it is glucose, it will become, glucose should also fit in this formula because this is also a carbohydrate. C6H12O6. So if you divide everything by 6, it will become CH2O. Correct?
Yes, sir.
Empirical formula. Remember? Have you heard it? Have you read it yet or not in chemistry?
We did, sir.
Okay. So you understand what I'm writing, correct? If there is any doubt, please ask. So this is the first thing that we got to know. Next, we also got to know that light is doing something. Now this light is doing something because of which this carbon dioxide is getting reduced to a carbohydrate, because in a carbon dioxide we already have carbon and oxygen, right? What we do not have in a CO2? Hydrogen. Do you understand?
Yes, sir.
So that hydrogen is coming from water. That reducing agent is coming from water, right? So write down. There was another person, write down. After this, a microbiologist named Cornelius Neal. So this is the fourth person you have to remember in this chapter. Cornelius Neal. Okay. Now, what did Cornelius Neal told was that, so he studied purple and green bacteria. Okay. And he showed that photosynthesis is basically a light dependent reaction. We already know that it's light dependent, but he showed that in which the hydrogen from any like oxidizable compound is used to reduce the CO2. So basically this thing he figured out, that this CO2 is reduced to glucose or starch, and in order to do this, or a carbohydrate, in order to reduce a CO2 into a carbohydrate, the hydrogen is coming from any substance that can get oxidized itself. So in chemistry, you know that whenever something releases hydrogen or donates hydrogen, it itself gets oxidized, correct? Correct? Yes,
Yes.
or no? Yes. Now if water gives hydrogen, then itself will get oxidized and it will become oxygen, because water has nothing than hydrogen and oxygen. So if it gives hydrogen, it's just oxygen, and oxygen is a oxidized form of water only, right? Because you remove all the hydrogen. Correct?
Yes, sir.
Making sense to you? Now write down in for Cornelius Neal. Cornelius Neal showed that photosynthesis is a light dependent reaction. Photosynthesis is a light dependent reaction in which hydrogen from any suitable oxidizable compound, which in this case is water. Hydrogen from any suitable oxidizable compound, write down in brackets, in this case water, is used to reduce carbon dioxide, is used to reduce carbon dioxide into carbohydrates. Into carbohydrates. Is it clear? So this was his contribution. His contribution is clear?
Yes, sir.
Yes.
Yes, sir.
Yes. Okay. Now write down the overall reaction of photosynthesis. Overall balanced chemical reaction of photosynthesis. Okay. So the reaction is simple. We have CO2. We have a suitable oxidizable substance, which is water in this case, coming from the roots. In the presence of light, we form glucose, C6H12O6, and we release oxygen, and also we release some water. So this water sometimes we all miss, but that's also there. Okay. Now, can anyone balance this for me? This is the complete reaction. Now balance this. So water is always taken by plants in excess, because this water which is coming from roots and this water which is going out from the stomata, remember transpirational pull, to maintain transpirational pull you have to remove lots of water also.
Yes, sir.
Yes. Now can someone balance this? Please tell me the balanced reaction.
Done, sir.
Yes, tell me. No, okay, don't tell me. Well, let me go to Omar. Omar, you're there? Will you speak today? Omar, can you hear me? Am I audible to you? If Omar will not speak, I'll go to Reba. Reba, you there? Can you speak? Reba, am I not audible, people? Okay, I think Reba or... Oh, Reba is speaking. Yes, Reba. Did you do it? Did you balance the reaction?
Yes, sir.
Yes. Can you tell me?
6 CO2,
Mm-hmm.
6 H2O,
Okay.
6 O6,
6 O2.
O2.
Okay. Is it balanced?
Yes.
No. See, hydrogen here is 12, but here 12 hydrogen are here only. So hydrogen is not balanced. Here are 14. Hydrogen is 12 here and 14 here. So this is not a balanced reaction. Anyone else would like to try? Or Iba, you yourself would like to try again? Aisha? What? I am having difficulty understanding Aisha. Yes, Aisha. Would you like to try?
Am I audible?
Yes, you're audible.
Shall I start? Is it?
Mhm.
Is it? Is it? Wait, so 1 minute.
What is the point of is it? Like you can just balance and know it for yourself. So you have to tell me what it is. You don't have to ask is it.
But so when you was asking me, I
You
was
cannot be wrong with calculations if you know how to balance LHS equal to RHS. You have been doing this in maths forever, right? Hence proved LHS equal to RHS. You just have to equalize You have to just follow conservation of mass here. Whenever we balance a reaction, what we are doing is we are following conservation of mass. So all the atoms of all the elements on each side should be equal, because nothing is destroyed or made in a reaction. Everything is transformed from one form to another. That's the concept of conservation of mass and conservation of energy in the universe, right? Is it clear? Except for like areas which are beyond like your study currently, like areas like black holes and Oort clouds where nebula and things like that, we follow conservation of mass everywhere in the universe. Yes. Who will tell me? Now it's an open. Who will tell me the balanced reaction? I'm testing
Second,
chemistry. Yes, Mariam.
6 CO2,
Mhm.
12 H2O,
Okay.
6 O2, 6 H2O.
6 O2 and 6 H2O. Is it balanced?
Yes,
Yes.
and 12H2. Yes, it's correct.
Yes, it's balanced. Perfect. So this is the balanced complete chemical reaction for photosynthesis. Okay, just write it down.
Actually, at that time, I
What?
was, like my voice was
Yeah, I
freezing,
can
and
I can
that's why I
understand.
wasn't
Because of your voice, you are not able to give a lot of answers. It's fine. And I think you are also struggling with network issues today. I hope my voice is perfect and you are understanding things. Okay, good. Okay, very good. So till now, everything is clear about photosynthesis, the pigments, what is the role of light, its spectrum, oxygen, glucose, everything clear? Okay, now let me go. So now let's go and try to figure out this photosynthesis happening in the plants. Now, so where this happens in the plant, remember I told you about chloroplast and chlorophyll?
Thylakoid.
What? Thylakoids. You used the word thylakoid, right?
Yes, sir.
What is a thylakoid?
A sac disc-like structure.
Okay,
which
let me ask you a
contains
few
the-
things. What is a plastid, a thylakoid, a chloroplast, and a chlorophyll? What are all these things? Anyone has an idea about this? In the cell, plant cell, you have studied plant cell versus animal cell, right? In class 10th. Say yes or no? So the
Sir, could you
organelle,
repeat?
I'm asking, there are many words: chlorophyll, chloroplast, thylakoids, and plastids. What are all these words means? So you used a word thylakoid. So Mariam, would you like to tell me what is a thylakoid?
Sir, it's a round structure which contains the pigment, chlorophyll.
Okay, it's a round what? Pigment.
Sac-like structure. Disc-like structure.
Okay, where is thylakoid present?
In plastids.
Then where are chloroplasts?
Inside the thylakoid.
Chloroplast is an organelle. See, this is the confusion. Okay, so what is plastids?
The organelle.
So let's first define these things, okay? Because these will create confusion otherwise. Okay, let's people start with plastids. Write down plastids first. Plastids. Write down. Plastids are membrane bound organelles. Plastids are membrane bound organelles found in, someone is asking something, thylakoid membrane. Okay, Aisha will come to it. Let's write about plastids first. Plastids are membrane bound organelles found in the cells of plants, algae, and some eukaryotic cells. Okay, so plastids is a group. There can be different kinds of plastids. Do you understand, everyone? Please be on the same page. Keep saying yes or no.
Yes, sir.
So plastids are membrane bound organelles. There can be different kinds of plastids, like chloroplast is a kind of plastid in plants. Okay, is it clear?
Yes, sir.
In other eukaryotic cells, there can be different other plastics called leucoplasts, amyloplasts, and proteinoplasts, which means amyloplasts are storing starch, chromoplasts are storing pigments which give colors. The word chromo means colors, and the word chloro means it's for chlorophyll, so it contains green pigments. So plastids are groups. There can be different types of plastids. Chloroplast is one type of plastid. So now let's come to chloroplast. Write down chloroplast. Chloroplast. Chloroplast is a type of plastid within the plant cells and certain photosynthesizing algae. Chloroplast is a type of plastid within the plant cells and certain photosynthesizing algae, or photosynthetic algae, sorry, photosynthetic algae is a good word, which contains chlorophyll, and is the site of photosynthesis. Clear, everyone?
Yes, sir.
Yes.
Now, what Mariam and Aisha both have mentioned, thylakoid. Now, what is thylakoid? Now, thylakoid is present inside the chloroplast. Is it clear, Mariam?
Yes, sir.
Okay, so write down. Thylakoids are membrane bound compartments. Yes, we also call it thylakoid membrane. So they are membrane bound compartments. So first thing is that chloroplast itself is a membrane bound compartment, and it's a double membranous compartment. Within that chloroplast also there are thylakoids which have their own membrane. So they are called thylakoid membranes. Is it clear? Is it clear, everyone?
Yes, sir.
Yes.
Yes.
So there is, and chloroplast are a type of plastids. In plastids, you can write examples: chloroplast, chromoplast, amyloplast, etc. And then we came to chloroplast. Now we are coming to thylakoids. So thylakoids are membrane bound compartments inside chloroplasts and cyanobacteria. They are the site of light dependent reactions of photosynthesis. In photosynthesis, I will teach you about two types of reactions. One is called light reactions and one is called dark reactions. Okay, so these thylakoids are the site of light dependent reactions of photosynthesis. Is it clear?
Yes, sir.
And thylakoid
Yes, sir.
and thylakoids have membrane over it, which is called thylakoid membrane. And write down one more line. The thylakoids in the chloroplast, are present in the form of stack of discs are present in the form of stack of discs. No, stack, you know the spelling of all, right? Stack of discs. Known as grana, known as grana, 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 whatever you want to call it. Okay, so these thylakoids are inside the chloroplast. Now, what is chlorophyll?
The pigment
Where is it present?
in thylakoids.
Yes, these are present. Write down chlorophyll. Chlorophyll is the green pigment which is present within the thylakoid membrane. Which is present between, which is present within the thylakoid membrane and the space between thylakoid and the chloroplast membrane. Is it clear? So it is
Yes.
also present in the within the thylakoid membrane itself, and it is also present between the thylakoid and the chloroplast membrane. And that region is called stroma. Write down stroma. So chlorophyll is present in stroma along with thylakoid membrane. Cool. See, all the four words are clear to you. Any confusion in any of these words now? No, I guess, right? No confusion. Shall I move forward? Okay, so let's now make a chloroplast just to represent it. So you understood that chloroplast is a kind of plastid which contains chlorophyll. That's why it's called chloroplast. Now chloroplast has an outer membrane and an inner membrane. Is it clear, everyone? Yes, so this is a chloroplast. This is the outer membrane of chloroplast, and this is the inner membrane of chloroplast. Now inside the chloroplast, I told you that there are thylakoid membranes. So they are stacked like discs, one over the other. Is it clear? Okay. And these are thylakoids, and this stacking is called as grana. Is it clear, everyone?
Yes, sir.
This stacking is called grana. And chlorophyll is present within, so I'm using green color to mark where chlorophyll is present. Okay, so chlorophyll is present inside also. Here, can you see this where I'm marking? And it is also present in granules between this membrane and the inner membrane. So this whole area is called stroma. Is it clear, everyone?
Yes, sir.
And then there are, of course, other things which I'm making in red. There are ribosomes present inside a chloroplast. Ribosomes are present, and then there are big granules which are called starch granules, because photosynthesis is happening here, so starch will be produced, so it has to be stored in some granules, right? So apart from grana, which is thylakoid membranes, stacked as a discs, and the stroma. These are the other things which are present. Is it clear, everyone?
Yes, sir.
Okay, very good. So I hope this is clear, and let's write something about the pigments first, which comes in entrance. Write down that I have already told you here. So this, all this graph you have understood. Let me just make you write certain things. Write down chlorophyll A. First, about chlorophyll A. Chlorophyll A. Write down. So if we okay, write down about chlorophyll A. It's the primary photosynthetic pigment present in almost all higher plants. It's the primary photosynthetic pigment present in almost all higher plants. So major pigment is this. Then we have chlorophyll B, which is the second most important photosynthetic pigment, chlorophyll B. Second most important chlorophyll photosynthetic pigment. And then we have carotenoids, and xanthophylls. These are important. Carotenoids and xanthophylls. So xanthophyle, like chlorophyll, xanthophyle are brownish, reddish brown in color, and they are reddish orange, or again like non-green. So these two are non-green pigments, non-green, non-blue pigments, okay, which plays important role in photosynthesis. So photosynthesis is not all about green pigment. Only chlorophyll is green. Chlorophyll A is green pigment. Chlorophyll B is, it becomes more bluish pigment. Is it clear, everyone?
Yes, sir.
Yeah. Mariam is asking, can you also define mesophyll cells? Like mesophyll cells of what? In what context you are asking mesophyll cells? Like in case of mesophyll that contains the chlorophyll?
Yes, sir.
Okay, so write down. See, mesophyll cells are basically the two types of cells which are found in the leaves, okay? So bulk of that leaf is filled with mesophylls. So they fill the bulk of basically the leaf, okay, every leaf. And these mesophyll cells contains chloroplasts, which then contains your thylakoids, in which there is chlorophyll. Okay. So the basic primary function of mesophyll cells is in photosynthesis and in transpiration. Is it clear?
Yes, sir.
Now we will be studying about mesophylls in detail. I will be covering it in this chapter, but for now just understand that mesophyll is the green bulk of the leaf. It's present in leaf. It and mesophylls contains chloroplasts, in which there are thylakoids, in which there are chlorophyll. Is it clear?
Yes, sir.
So you can write that mesophyll cells
Yes, sir.
are found in leaves that contains chloroplast and are necessary for photosynthesis. That could be the definition for now. We'll come into detail of it. Okay. Is it fine for now, Mariam? Shall I just
Yes, sir.
move ahead for now? Okay. So if someone asks how many type of pigments are there, you know that there are both green, green, blue, and non-greenish blue pigments as well, right? Chlorophyll A, B, carotenoids, and xanthophylls. These are all the pigments. Is it clear? Now write down
Yes,
about
sir.
some basic things about photosynthesis. I'll dictate and you will write now. So in a chloroplast, basically what happens is there are enzymatic reactions that happen which synthesize these carbohydrates and starch. Okay, so write down in photosynthesis. In photosynthesis, there are two types of reactions majorly happening. Okay, that's important. So in photosynthesis, there are majorly two types of reactions that are happening. The first kind of reaction, this will come under types of photosynthetic reaction. Types of reactions in photosynthesis. Okay, so first will be called photochemical reactions. Photochemical reactions, or in common terms
अल्लाह के नाम पे जो कोई मुझे नेट दे दे, पूरा टाइम रिकनेक्ट हो रहा है, गुस्सा आ रहा है मुझे.
Fine, it's fine. मिल जाएगा. Okay, so the first kind of reactions are called photochemical reactions, which in normal terms are known as light reactions. And they are called light reactions because they happen in the presence of light. Is it clear? And what do they do? Write down. Write down. Light reactions are responsible for trapping the light energy. Light reactions are responsible for trapping the light energy and also for the synthesis of trapping of light energy and also for the synthesis of ATP. What is ATP? ATP, what is ATP?
Adenosine triphosphate.
Adenosine triphosphate, which is the energy currency, right, of the cell, and NADPH. Just write it down. I'll explain what it is later. So the first set of reactions, two kinds. So this happens majorly during the daytime. So when there is day and lot of light you can gather, so photosynthetic machinery mostly does this, the photochemical reactions or the light reactions, where they trap as much light energy as they can and then they synthesize ATP and NADPH. Okay. Is it clear?
Yes, sir.
The second type of reaction which is followed, so first this happens. The second type of reaction is called carbon reactions. Carbon reactions, or just opposite to it, they are called dark reactions. But the catch here is that these reactions do not only happen in the dark, they are just not dependent on light. Is it clear, everyone? So you should not think that half of it is happening during the day and the other half is happening during the night. You know?
Hey, I'm sorry, I got disconnected.
Okay. No problems, Aisha. I'll just repeat. I was just telling that there are two types of reactions happening during photosynthesis. The first type is called photochemical reactions or also called light reactions or light dependent reactions, because they are dependent on light. So clearly they can only happen during the daytime. Okay. In these reactions, trapping of light energy happens and synthesis of ATP and NADPH happens. Is it clear? The second type is called carbon reactions, or just opposite to it, like by convention they are called dark reactions. But this does not mean that they happen in the dark. This means that they are just not light dependent. So write it down. They are not light dependent. So they can happen anytime. Is it clear? During the day also and during the night also. Is it clear, everyone?
Yes, sir.
So if the question in entrance comes that which reactions happens throughout 24 hours in the photosynthetic pathway, then which reactions happens throughout all the time? Dark reactions. Okay, Mario?
Yes, sir.
Yes. Dark reactions does not mean that they happen in the dark. It means that they are not light dependent. So they are happening during the day also and during the night also. But which reaction can happen only during the photo period time?
Light reaction.
Light reactions, because they are light dependent. Photochemical reactions can only happen during the daytime. So in the night time, it's not like photosynthesis is not happening, but only the second type of reactions of photosynthesis are happening in the night, because in the daytime both are happening. Is it clear?
Yes, sir.
Now write down for the second type, which are carbon reactions. These reactions synthesize sugar. These reactions synthesize sugar, which in turn forms starch. So synthesis of sugar, which is glucose, which in turn forms starch, the stored part, the stored substance, stored form of sugar. So do you understand that there are two types? There is a division of labor. There is a clear-cut division in how it operates. One operates only during the day and the other operates both during the day and the night. So you will not get confused by the name light or dark now. Is it clear?
Yes, sir.
Okay, very good. Very good. So now we have to just understand in detail what is the light reaction and what is the dark reaction. And we also have to understand one thing called electron transport, where I will tell you about the NADPH and electron acceptors and electron donators. So for that, and what we are going to cover in the next class, which is tomorrow, is mostly lots of chemistry based. So I'm not starting it today because we have just 5 or 6 minutes left, but I'm giving you assignments, and please do this very religiously, follow it strictly, because tomorrow otherwise you will struggle in the classes. And also make sure your networks are good, at least your audio bandwidth is good. So what you have to go and do today is you have to read about what is electron donor, just the basics in chemistry, and electron acceptor. Okay. Did you heard something? Yes, Aisha, we hear you were begging for internet, which is a good thing. It's fine. So electron donor and electron acceptor you will read about. Okay. Is it clear for all of you? Say yes or no?
Yes, sir.
Because I will be teaching you the electron transport system in plants, which is the most crucial part of photosynthesis, from which lots of entrants, like in every entrance, at least 2-3 questions are like, you just take it for granted that they are coming from this part. Okay. Electron donor, electron acceptor, and also try to figure out what are cytochromes. Just read basics of it, cytochrome and phosphorylation. These are some chemistry terms. Phosphorylation. These 3 things you read and come tomorrow. Is it clear?
Yes, sir.
please do it. No one will come in tomorrow's class before doing this. So in
Okay.
tomorrow class we'll be doing the major bulk, which is very entrance heavy stuff. So please come prepared. And of course, after all this, plus revise everything again. Revise today's class thoroughly. Okay.
Yes, sir. And I'm so sorry for today. I wasn't knowing.
Right, it's fine, it's fine.
Okay, guys.
Okay, so see you tomorrow, and tomorrow I'll be teaching you all this that I told you.